Hi everybody, we're going to pick up with muscular system excitation contraction coupling. Now this information begins in your course pack on page 83. I highly recommend that you spend time reviewing this PowerPoint and the information in your course pack because this is really the first encounter we've had with a thick physiological topic. So you'll want to make sure that you have ample time to go back and learn this as a story. This is a stepwise process, and if you can repeat the steps without having to refer to your notes or your book, then I think you have a comfort level that will do you well on the test. So excitation contraction coupling is just fancy language for how we tell a muscle cell from a nerve that it's time to contract. Excitation involves a neurotransmitter, which is a chemical messenger that travels from a nerve cell right next door to a muscle cell. That neurotransmitter stimulates the muscle cell and causes excitation. Keep in mind, excitability is a term we defined a little earlier in this unit, and excitability means that a cell can respond to or sense a stimulus. Now, what follows is a contraction event. Contraction involves the skeletal muscle contracting and shortening. That occurs in response to excitation. You shouldn't have contraction unless you have excitation first. We'll talk through the sliding filament theory, and I've also pulled that information out and placed it in a separate PowerPoint slideshow for you. So do take a look for our muscular system sliding filament theory narration. In order to understand excitation, we need to talk about two terms here, motor units and neuromuscular junctions. Motor unit consists of a neuron and all the skeletal muscles that it stimulates. In this picture, the motor neuron stimulates this entire bundle of muscle cells. So the entire picture would be classified as a motor unit. Now, you'll notice this motor neuron has little branched endings. These branched endings are called neuromuscular junctions. Literally translated, neuromuscular junction is the place where the nerve and the muscle individually join together. So I have circled here one individual neuromuscular junction. Here's another and another. We could keep circling these. And they are the place where the nerve and the muscle come together. Now, they don't actually touch one another. There is a gap of space between that nerve and that muscle cell. That space is called a synaptic cleft. In order for you to see that synaptic cleft, I'm going to have to turn this picture sideways. So again, this is a neuromuscular junction. It's part of a larger motor unit. When that neuron sends a nerve impulse to those muscle cells, they should all contract together as a unit. That's of course why they call this a motor unit. But where the nerve and the muscles individually meet is where we call this specific junction a neuromuscular junction. Here again you can see individual muscle cells. They're traveling across the screen this way. And here we have the axon terminal. That's that little foot-like ending there attached to an axon and to a motor neuron. These are muscle cells here traveling across. And each of these are individual axon terminals, and they sit on or nearly touch an individual muscle cell. The junction is created here. This is called a neuromuscular junction, where this axon terminal nearly comes in contact with that underlying muscle cell. Like I mentioned, they don't actually touch one another. There is a little bit of space between the two. All right, so yet another look at that neuromuscular junction, and now let's turn it sideways so you can see that synaptic cleft. Here you can see the synapse or synaptic cleft is a gap of space sitting between the axon terminal and the nearby muscle cell. So if you zoom in and take a look, the sarcolemma, you may recall, is the plasma membrane of that muscle cell. Okay, so that's it right there. And this below is not actually labeled, but it is the muscle cell. And of course what we have here, this piece diving in, I'll just put a label over here, that's the T-tubule. That's a piece of the sarcolemma that takes a dip down into that muscle cell. So now that we have this turned sideways, we can appreciate that there is a bit of space between the axon terminal of the nerve cell and the neighboring muscle cell. That gap of space is called a synaptic cleft. That synaptic cleft forms part of a synapse, which is this unit between the axon terminal the gap, and of course the neighboring muscle cell. All right, so let's talk about how we transmit nerve impulses from nerve cells to muscle cells. That takes us to this picture here called excitation. So let me walk you through the process. In order for a nerve impulse to be transmitted to a neighboring muscle cell, we have to relay the message. Well, since this nerve cell isn't going to tap or bump that nearby muscle cell, we have to figure out a way for those two cells to communicate. And we do so by the release of a neurotransmitter, which is a chemical messenger, kind of like a hormone, but this one doesn't travel the bloodstream. This simply has to travel across that very small synapse. So in order for us to transmit the nerve impulse via neurotransmitter, we have to initiate the process with step 1. So in order for us to relay the nerve impulse, calcium has to enter into the axon terminal of that nerve cell. So calcium rushes in, and that allows for us to prepare vesicles full of neurotransmitter. If you don't have calcium rush in, the next step will not follow suit. So these neurotransmitters we're going to discuss are called acetylcholine, or ACH for short. So we're just going to refer to it as ACH from this point forward. That's what are these neurotransmitter molecules. So this particular neurotransmitter, ACH, travels in these vesicles toward this end here of your axon terminal. When we have these vesicles fused with the membrane here, they will basically spit their guts out. They rupture just like a blister, and those ACH molecules now escape into the synaptic cleft. Keep in mind that is the gap of space between axon terminal and neighboring muscle cell. That acetylcholine travels a very short distance across that gap and binds with a protein channel. Now this protein channel is normally closed, but when acetylcholine binds, it's like putting a key in a door. It allows us to open up that protein channel. So that's what we see over here. This neurotransmitter shown up here is ACH. You can see that it binds to that protein channel, and when it does so, that sodium ions can now travel into that cell. So this is not just any old protein channel. This is a protein channel that's specifically for sodium. So we can say it's a sodium channel. Now, in order to unlock and open this channel, acetylcholine must bind to that channel. So as long as acetylcholine sits there, sodium rushes into that muscle cell, and that causes excitement or excitation inside that muscle cell. We'll talk further about excitation in the next chapter when we discuss nerve impulses in the nervous system. So for now, we'll just know that sodium rushing in changes the environment from a negative environment to a positively charged environment, and that's what causes excitation inside that muscle cell. So if you don't want to have continued excitement, you have to get rid of acetylcholine. It's kind of like breaking the key once it's opened the door. There is an enzyme that comes along to break down this neurotransmitter. It's spelled ACHE, or acetylcholinesterase. Did you catch the ending there? Acetylcholinesterase has ASE as an ending. That's because it's an enzyme, and it comes along and it breaks down acetylcholine into acetyl and choline. Okay, so we can actually recycle these components and put them back up into the axon terminal for another go-around. So really cool thing here is that we don't waste the acetylcholine. We can recycle and reuse it. All right, so what does ACHE do for us? Well, in breaking down acetylcholine, it prevents further unwanted muscle contractions. Remember, as long as acetylcholine sits on that sodium channel, what happens to the sodium channel? It stays open. And what comes through that sodium channel? Are sodium ions. And when sodium ions rush into your muscle cell, what happens to the environment in there? It becomes positive. So it's that change to a positive environment that causes the excitement that we need to promote the next step, which is contraction. So acetylcholinesterase is important in breaking down ACH. Why? It prevents further unwanted muscle contractions. All right, so the whole goal here was to allow acetylcholine to open up a sodium channel The sodium channel allows sodium to rush into that muscle cell. That temporary permeability to sodium is short-lived because ACH will, of course, break down and remove acetylcholine. In doing so, you notice the bottom picture, that channel closes up on us. All right, so a nerve impulse here is now generated inside this muscle cell. We call it an action potential here. And this wave of excitement is conveyed by those sodium ions now that are inside that muscle cell. This now allows us to kind of continue the conversation and talk about contraction. But first, I'd like to help you look at the picture on the top of page 84 and label it. So let's take a look at this picture on the top of page 84. First of all, you have to initiate the process of getting the nerve impulse transmitted from the axon terminal, which is this structure here, to the nearby muscle cell. So let's go ahead and label axon terminal, and this down below is, of course, our muscle cell. The sarcolemma is another name for that plasma membrane. Okay, and let's go ahead and talk about the other things that we see here. We have vesicles, and they are full of a neurotransmitter. Do you remember the specific type? ACH, or acetylcholine. Okay, we also have calcium up here, and these are sodium ions down here. Now, these are sodium channels, so let's go ahead and label those. All right, so along comes a nerve impulse. It travels down this axon terminal. I'm going to go ahead and label that as our action potential or nerve impulse. Same thing, okay? And that action potential triggers opening of calcium channels. And I'm just going to draw one right here. It's going to be like a little doorway. And so these calcium ions can come into this cell. And they travel through that open calcium channel, and they help to trigger the release of these vesicles full of neurotransmitter. Those vesicles are going to rupture like blisters, and the neurotransmitter ACH is out here now in the synaptic cleft. We know that ACH travels over here and binds to a sodium channel, and in doing so, it causes the sodium channel to now open. So that's important because ACH regulates the opening of that sodium channel. Now that sodium channels are open, sodium may rush into the cell through these open sodium channels. Okay? And that changes the environment in here from a negative state to a positive state. And that positive state is going to cause excitement. And the excitement is what we need because that tells the muscle cell, hey, it's time to contract. So let's do this one more time. In order to promote excitation in the muscle cell, the first thing we need to have happen is the nerve impulse arrives in the axon terminal, triggering the opening here of these calcium channels. Calcium rushes into this axon terminal, and that allows for these vesicles full of neurotransmitter to move to the edge here and rupture like blisters. So ACH now is present in that synaptic cleft. Acetylcholine travels over and binds to one of our protein channels, happens to be a sodium channel. Normally it's shut, but ACH is like a key. It causes that sodium channel to open. Now sodium can rush through. So the entry of sodium into our muscle cell causes a wave of excitement. It changes the environment from negative to positive, and that excitement is what leads to our next step, which is the contraction of that muscle cell. Let's now turn our attention to contraction. The sliding filament theory explains how muscle contraction occurs. This information is found in your course pack on the bottom of page 84. So this picture here is something we've looked at before. When the wave of excitement comes with sodium ions in the muscle cell, that wave of excitement causes the sarcoplasmic reticulum to release stored calcium. Now, when calcium exits the sarcoplasmic reticulum, that's a sleeve around your myofibril, the intracellular levels of calcium now begin to increase. So we tend to see calcium in the sarcoplasm increase and the sarcoplasmic reticulum levels begin to decrease. That's because calcium is heading to these actin filaments and binding to the troponin complex. What happens to that rope-like tropomyosin when calcium binds? Well, that rope-like tropomyosin is now moved out of the way, and it exposes myosin binding sites on these actin filaments. These binding sites here are going to be the place where myosin heads can attach and promote a muscle contraction. In this image, you're actually watching myosin heads walk along and form cross bridges and power strokes with the thin filament. So this myosin head is binding to actin binding sites. Well, technically they're for myosin, right? But they're only actin filaments when those ropes of tropomyosin move out of the way. So how do you move that rope of tropomyosin? Calcium has to bind troponin. Where does the calcium come from? Calcium comes from the storages in those sarcoplasmic reticulum. By the way, where are the sarcoplasmic reticulum found? They loop around those myofibrils. Okay, so every time that myosin head comes in contact with an actin filament, we say a cross bridge has formed. You'll notice immediately after making contact, that myosin head begins to pivot, forming what we call a power stroke. Now, that power stroke is what enables us to move or slide or pull along that thin filament. Keep in mind, the big picture here is the name of this process is the sliding filament theory. So in order to pull or slide those thin filaments, we use the myosin heads to pull and move along these thin filaments. Neither the myosin nor the actin filaments shorten here. All we're doing is sliding those Z lines closer together. So take a moment to study all the components of this picture. I have broken this one down in more detail in our separate narration on the sliding filament theory. So here's a picture of a relaxed muscle. You'll notice that there are I bands and H zones present. That is what I need to know if I'm going to say this is a relaxed muscle. So you've got a space here in between those thin filaments right there where there's no thin, there's only thick, and you've got a pretty sizable I band. So that's confirming for me that we have a relaxed muscle. As the muscle begins to contract, these myosin heads reach up and bind to these thin filaments, and the thin filaments are being pulled toward the center of the sarcomere. So simultaneously, heads are binding, forming cross bridges, power stroking, releasing, and doing this all over and over in order to pull and slide those thin filaments toward the center. Over time, you can see here that we lost our H zone. It should have been right here through the middle, but in fact, we have bunched up our thin filaments as they overlap, being pulled toward the center of the sarcomere by those myosin heads. What's happened to your I bands? Well, they're pretty much nothing. You know, you've got Z lines here, and you don't have any space on either side where you could qualify that as an I band. So the I bands are disappearing, the H zones disappear, and what happens to the overall size of this sarcomere? It shortens. So you can thank those myosin heads performing this walking structure, this power stroking to help pull and slide those thin filaments. Let me make a very important point here. Neither thin nor thick filaments themselves will shorten. All we're doing is pulling and sliding filaments to cause the muscle's sarcomere to shorten. Keep in mind the sarcomere is the distance here between these Z lines. So there in fact has been no shortening of any filaments, just a sliding process. This image also shows us what happens when a muscle contracts. I think you can easily spot our Z lines right here. Okay? And as this muscle contracts, the Z lines do move closer together. And of course, we see no more H zone in the middle, and the sarcomere has shortened. So that kind of confirms what we think about what happens with muscle contraction. It is the sliding of filaments past one another. So let me walk you through the process of excitation contraction coupling one more time. We're going to start with excitation. Excitation occurs when neurotransmitters can be released from the axon terminals. Do you recall what must enter into that axon terminal for us to be able to release neurotransmitter? It's calcium. Okay, so calcium must come into that axon terminal in order for acetylcholine to be released. Once acetylcholine binds to sodium channels on the sarcolemma of the muscle cell, those sodium channels open up, and of course sodium rushes into the muscle cell. So that sodium was hanging out in the interstitial fluids between your axon terminal and this muscle cell. Here's a T-tubule right here, and you'll notice that action potential can be carried deep into the inner portions of this muscle cell. So by doing that, that takes this nerve impulse right there to those sarcoplasmic reticulum. That's what this structure is. Well, we know the excitement of having sodium inside a muscle cell leads us to our next process, which is contraction. We know the sarcoplasmic reticulum stores calcium. Here's yet a second time we need calcium. When calcium is rushing out of your sarcoplasmic reticulum, it's doing so to increase the amount of calcium in your sarcoplasm. The more calcium you have in your sarcoplasm, the more likely it is that we can perform a muscle contraction. Here you can see that calcium comes into your troponin complex right there. It binds that troponin complex, and what happens next? This rope-like tropomyosin right there will now move out of the way. It's like a blindfold, and when it moves, it reveals binding sites on those actin filaments. When that happens, this myosin head right there can reach up and form a cross bridge, and then power stroke. Power stroking will cause this whole thin filament to slide. Sliding is part of that sliding filament theory of muscle contraction. In order for this head to release and repeat the process again and again, we have to power the head's release using ATP. So repeatedly, this head binds, forming a cross bridge, pivots, forming a power stroke. That power stroke is what pulls and slides that thin filament. ATP comes along, it powers the release of that head, it lays back down into a relaxed position, and then if those binding sites are still available, it comes back up again, binds, and then pulls that thin filament. So this repetitive process creates sliding and contraction and shortening of that sarcomere. So once you're done with this process, we can then store the calcium back into the SR. You know, if you're no longer performing a muscle contraction, there is no need to leave those binding sites on the actin filaments exposed. So those ropes of tropomyosin will of course cover up those binding sites. Calcium here is stored back up here in the SR. Well, if you cover up those binding sites, then this myosin head no longer has anything to attach to, so ATP once again powers its release and it comes back into its relaxed position. Unable to cause that muscle to slide and shorten, the muscle relaxes and then goes back to just a resting state. All right, so this actually concludes our process. When you've put everything back to a resting state, then we now can say that that muscle has gone through its contraction cycle and then now reached back into its relaxation state. So here you can see the binding sites are covered by the rope-like tropomyosin. The calcium is stored back in the SR. This myosin head now is going to relax back to its original shape, and of course that muscle is going to lengthen and the sarcomere will spread back to its normal situation where those Z lines have some space between them. Just a little bit left on this page. This is 85. Some muscle contractions can occur with shortening, some do not. I just want to introduce to you that there's two types here. Muscle contractions that shorten and produce movement are isotonic contractions. That's what you normally do when you move about. If you bend over and pick up a pencil off the floor, that's an isotonic contraction. Flexion, extension, abduction, and adduction are all isotonic examples. Now, you sometimes perform isometric contractions. Since both of these terms have iso as a prefix, I find it helpful to look at metric as length or meter. So isometric contractions do not change length. In this case, you might be leaning against a wall. For instance, if you take your outstretched arms and push against a wall as hard as you can, you'll feel your muscles tighten That's when the myosin heads bind to those binding sites. But you're not going to be able to push the wall down. So you only feel the muscle tense up, but you can't shorten that muscle. So isometric contractions occur when muscles are trying to shorten but are unable to do so. So there is contraction there in the aspect that the myosin heads bind to the thin filaments, but they're not able to pull and shorten the muscle. So here you can see how an isotonic contraction actually causes muscle shortening. But by contrast, an isometric contraction does not cause the muscle to change length. Those myosin heads will bind. You have tension and, what you feel like is potential energy built up in that muscle, but there's no shortening that occurs here. Just want to mention at the very bottom of page 85 that smooth muscles do contract. Of course they do, right? They're involuntarily controlled though, and their process is actually peristalsis. That is a rhythmic contraction that produces long-term contractions without getting this muscle tired. So the process here is a little different since we're missing the striations that we normally see in the skeletal muscles. We're not going to go into detail about how smooth muscle contracts. I just wanted to remind you that peristalsis is the name of the muscle contraction that occurs here. You can see that peristalsis is a rhythmic contraction that involves contractions and relaxations. This is in fact how food is moved along our digestive tract from mouth to anus. It's also the way that we can move blood through our vessels, urine through our urinary tract. So it's a common type of process used by smooth muscles in the walls of these hollow organs.